So just to then kick off, we're going to start with Cheminformatics. So Cheminformatics is all about chemical information and capturing chemical information and representations of small molecules on a computer to be able to then manipulate and retrieve, and then ultimately in generative design be able to hopefully build novel chemical structures which we can then have made in the lab. So Cheminformatics is really concerned with sort of that foundational representation. So starting then to think about individual molecular representations. Molecular representations are interesting because there are many different types. However, some foundational ones that will appear often in generative design can be seen here. This comes from this wonderful paper, and I just want to step through each of them briefly. So the first one is going to be familiar, I think, to everyone. This is the depiction. So in a depiction, this is where we draw a chemical structure and we store that chemical structure in terms of a sort of a pixel representation on a plane. We store additional chemical information such as the stereochemistry of each of the sort of centers. We store approximate sort of 2D spatial layout of the chemical structure, and we encode this as a collection of pixels. Now, the important thing to remember about depictions is that they are not molecules themselves. They are just the way that we draw them in 2D space on a plane. However, they are a bit cumbersome to be able to use. So instead, often what we will do is we will instead prefer to represent a molecule as what we call a chemical graph. So a chemical graph is where we take each atom and transform it into a node, and we take each bond and transform it into an edge. And what this chemical graph allows us to do is that this is a sort of mathematical object which we can then begin to transform. So we can apply transformations to this molecule. And by doing that, we then can add atoms, we can delete atoms, we can bind on fragments, we can do quite a bit of work. And in fact, all sort of major cheminformatics packages will rely upon the chemical graph as the underlying representation. However, a limitation of the chemical graph is certainly that it can be quite bulky to store. So instead, often we will store the chemical graph as a smile string. So a smile string is a spanning tree over the molecules. So it's where we start at a particular node, and then we can traverse the molecule, and we then can actually write out the individual bonds and the rings and the stereochemical information. So this is is a representation of stereochemical information, and so is this double at here. The key takeaway of this representation, however, is that because we can start our traversal from any node that we like, usually we pick a heavy atom for this, what that means is that we can actually draw smile strings, or we can write smile strings in multiple different ways. So a single smile string is not necessarily the canonical description of the chemical graph. To do that, we have to do a process known as canonicalization, where we sort of universally force the traversal to always happen at a particular node. However, smile strings overall are very convenient as they are string-based representations, so we can store many molecules inside a database, and it is relatively easy representation to handle. One then extension is to then start to think, okay, well, we have 2D graphs here. How do we then go and extend this to 3D graphs instead? So this is a 3D ball and stick based representation. This now starts to encompass what we call explicit hydrogens. This is where we start to actually encode the hydrogens, because the hydrogens will influence the 3D geometry of the molecule. And with this, we then start to get a spatial understanding of where the atoms and bonds are positioned. The next and sort of final representation, which we will see in generative design, is where we start to then think about adding a mesh over the top of that 3D structure. So what we do is we try to then encompass representations which include information around the electron density, and that really then allows us to start to use this representation to predict additional, more complex properties. It is, however, very computationally expensive to calculate, especially if we are generating many thousands or hundreds of thousands of molecules. Okay, I really want to sort of explore this idea of molecular graphs in more detail. And I think the reason why that is that cheminformatics really relies upon molecular graphs and how to manipulate them, and thus generative design, when thinking about atom-based, fragment-based, reaction-based, or different types of scoring and search, relies upon graph representations. So having a firm understanding now will help us later on. So first of all, sort of this figure here from this paper shows, I think quite nicely, how we transform a depiction into a 2D graph and a depiction in 3D into a 3D graph, and what are the sort of important aspects that we can capture. So here is aspirin in 2D. It is a depiction, so it is a drawing of what we would expect aspirin to be, and we can take each atom and transform it into a node, and each bond we can transform into a corresponding edge. And what we have then is this 2D topological graph. And what is a topological graph? Well, topological really is a fancy word to describe the sort of connectivity, relationships between the nodes in the graph, the nodes and the edges. And the key thing here with the 2D topological graph is that we're not defining explicitly what the 2D coordinates are in space. So we're only worrying, for example, that these this carbon atom is connected to this oxygen, it's connected to this carbon, it's connected to this carbon. So that's the key thing. So when you see the word topological, this really means connectivity relationship, in absence usually of distance information. So the 2D topological graph is actually quite a rich structure. So we can see here that we have nodes, and we have edges, and we have different colored nodes. These are different atoms, and then we have different colored edges, and each edge then encompasses a different bond type. So bond types are important because bond types allow us to get an understanding of the actual connectivity as related to chemical structures. So for example, we have double bonds, we have single bonds, we have aromatic bonds, and this forms then the overall graph. And that again contains a rich amount of data, a rich amount of information that we can then use to be able to build better molecules in generative design. The next sort of extension to this is then when we start thinking about representations in 3 dimensions. Now, in 3 dimensions, we suddenly have to worry about the interatomic distances. So here is the ball and stick representation of aspirin in 3D, and we can again take each atom and each bond, and we can then build, in this case, a 3D geometric graph. And this is a 3D geometric graph projected, importantly, onto a 2D plane, and it's termed a geometric graph because we start to worry about the geometry of the molecule. We worry about the interatomic distance, so we can see here that we have an additional sort of edge information. We have the distance between nodes here, and we can of course combine the 2D topological graph with the 3D geometric graph. However, it's important to note that this starts to encompass 3D information which the 2D topological graph usually would not. So molecular graphs are very, very useful to be able to store and manipulate chemical information. So Moving on from chemical graphs, we can now start to think about how do we store them. And so one of the key things is that storing a chemical graph is a bit tricky. So we could store the connectivity between each of the atoms. So we could store the information inside the table that this oxygen is connected to this carbon is then connected by 2 bonds to this oxygen, and it's connected to 1 bond to this carbon is then connected to this one, and so on and so forth. However, it is much easier to instead rely upon string-based line notations to be able to store our chemical structures. And 2 key sort of representations, line notations that are often used are SMIRKS and InChI. So SMIRKS, as mentioned before, is a spanning tree. It is a traversal over the chemical graph. So this is where we start at an atom and we traverse the graph. And often when we're storing SMIRKS, we can store many hundreds of thousands of SMIRKS because they are just simply string-based line notations. And this means that they're a very attractive representation to use in cheminformatics, and certainly in generative design, when we are storing the output of our generative design, we will usually use the SMIRKS string to do that. And of course, they do come with issues such as where do we traverse from, how do we canonicalize, and so on. On the other hand, we may often see InChI, and InChI is a different type of representation. So InChI is a layered representation, and InChI stands for International Chemical Identifier, and it effectively is a set of layers of different chemical information which encompasses all the information that this chemical graph contains. And so you capture information about the stereochemistry, the chemical formula, the connectivity of the carbons and the hydrogens, and where they are positioned on the molecule. And one of the things about InChI is that every molecule should resolve to a unique InChI. You can also, alongside InChI, store the auxiliary information. So this then encompasses a significantly larger amount of information. I'm not going to go through this in detail here. However, there are some limitations with InChI, and that is that InChIs, whilst they are unique and can be used for deduplication, to make molecules unique, they will instead transform the molecule into a sort of fixed representation. So they will take an opinion on what the tautomeric state of this molecule is and then write out that molecule in what they believe that tautomeric state is. So if, for example, we have molecules which we have explored at perhaps a different pH, then we may end up generating an InChI that doesn't resolve back to the chemical structure that we are exploring. That is, they can end up with different tautomers. And this can also lead to tautomer collision using the sort of InChI identifier. Now, one of the sort of final things I want to say is that regardless, certainly SMIRKS and InChI, they are used a lot. InChI is used in deduplication, and SMIRKS is where we store our molecules on output in a compact and friendly representation. So thinking then past sort of string-based representations also appear when we want to start storing chemical reactions. So chemical reactions are slightly different than regular sort of molecules, and that's because we usually have to deal with multiple products, multiple reactants as part of our chemical reaction. And so, for example, here we have the left-hand side of the reaction arrow. This is our starting materials, and then our right-hand side is our products. And we see all of these numbers. This is where we have then mapped all of the atoms from our starting materials onto then our products. And the key thing here is that this sort of atom mapped reaction is one reaction example, and we can store that one reaction example into this atom mapped reaction SMIRKS representation. And then we can build the InChI, and which is the sort of reaction international chemical identifier, following a similar process to the InChI as described on the sort of previous slide. We also can have InChI keys. I'm not going to go into these too much. However, there are other representations where we try to store individual reaction examples. Then moving across, we can then start to think about, okay, how do we genericize, how do we make a generic reaction for, say, an esterification reaction or an amide coupling or a Suzuki reaction? How do we achieve that? And what we can form is this thing known as a SMIRKS. And a SMIRKS is where we only try to encode the changing parts of a reaction. And this means that we capture the reaction center and how it is transformed. So what this will then allow us to do is this will allow us, for example, in reaction-based design, allow us to prospectively transform molecules successfully based upon the SMIRKS transformation that we can encode. Another representation, I'm not going to dwell on it too much, is the condensed graph of reactions. So this is where we try to store a sort of transition state of a molecule. This condensed graph of reaction can then form a graph signature, which then again can be stored successfully and used prospectively to be able to transform molecules. Finally, I'm going to highlight, I think, probably one of the most important parts, certainly for scoring when we think about cheminformatics, and that is descriptors. And so descriptors fall into two categories, really. They are really how do we calculate properties associated with molecules. So we have classical descriptors and we have data-driven descriptors. In classical descriptors, we are thinking then very much about things such as molecular weight or counts of nitrogens. These form what we call zero-dimensional descriptors. From there, we can then start to look at topological descriptors, so the connectivity between individual atoms to try to form substructures, which are then stored in fingerprints. You may have come across fingerprints before, so for example, the ECFP fingerprint, the Morgan fingerprint, the Max fingerprint. These are all sort of formed around the idea of a one-dimensional topological substructural fingerprint. Then we have classical descriptors. This is where we take our chemical graph and we store it as a distance matrix. We can extend that into 3 dimensions, again forming a matrix. So we can see here these are both sort of matrix-based representations. And then finally we can begin to store additional binding information over a mesh, and that will then form a 4D tensor-based representation here. A kind of new approach is then thinking past this and thinking then about data-driven descriptors. So in a data-driven descriptor, this is very much using, for example, a neural network where we attempt to encode in a latent space molecules, and then we use that latent space to then decode from that sort of initial vector in the center here to our chemical structure. And what we do when we go from encoding to decoding is that we somehow capture the chemical space and how we represent molecules in the middle here. And that's very attractive because we can use both encoders to be able to encode molecules, to be able to do perhaps machine learning, but we can also then use the decoder to be able to transform that descriptor back into a chemical structure. So these are known as data-driven descriptors because certainly the latent space depends upon what we put in, but there is another type of data-driven descriptor, and that's where we start to now think about capturing a description of a molecule based upon, for example, multiple angles associated with the chemistry or target information or network biology or cell biology and cell information or even clinical information. So this is where we start to push past sort of our understanding of a molecule in terms of the cheminformatics, and we start to pull in additional chemical and biological information to form data-driven descriptions of our molecules.